In this video, we will discuss how to critically analyze clinical data and introduce basic concepts related to the FDA regulatory process. And we'll do this by continuing the story of Biogen's aducanumab. So last video, we discussed how Biogen lost $30 billion worth of market cap because they announced a key phase 3 program in Alzheimer's disease failed. And in this video, we're going to discuss how they regained some of that market cap. Basically, they announced new clinical data, and the market rewarded them by giving them an increase in valuation. But we're going to critically evaluate that clinical data and see whether we agree with that or not. So we always need to analyze data critically because companies are generally incentivized to make their data look as good as possible. And it's especially important in this case. So Biogen is planning to file a regulatory filing for its drug in Alzheimer's, despite announcing 7 months earlier that they were canceling the program because the drug didn't work. So before we analyze the data and the implications of this announcement, let's just take a step back and just define the basic terminology here. So they're submitting a regulatory filing for aducanumab in Alzheimer's disease. And then later on in the press release here, it says they're submitting a BLA, a biologics license application. This is a formal submission to FDA asking them to approve a drug for marketing. So if you're not familiar with the drug development process, before a drug is FDA approved, you cannot legally sell that drug. You cannot legally market that drug to help treat patients with the disease. So getting FDA approval is a huge milestone for a drug, accumulation of years of R&D, and it potentially opens the door to the first profits ever in a drug's life cycle. So just as a quick side note, you'll also see NDA, new drug applications. So these are basically analogous processes. These are both submissions to FDA for regulatory approval. The difference is that biologics license applications cover large molecules, biologics like proteins, whereas NDAs, new drug applications, cover small molecules, which are smaller chemical structures, usually produced through chemical synthesis rather than through biological processes. So these are incredibly detailed submissions or scientific packages, literal truckloads of information in the days before online submissions. And these applications are prepared in accordance with FDA guidance and contain all the data that FDA is going to need to determine whether a drug is safe and effective for treating the target patient population. So why would Biogen go to all of this trouble to submit for FDA approval if the drug had failed? So it seems like either they made a mistake in declaring that the study failed and the drug actually works, or maybe they're not sure if the drug works or not, but they think it's worth it to just submit this application based on the overall risk-reward trade-off for this drug. So we'll look into both of those cases in the rest of this video. But first, let's just discuss why they stopped the study, and then we'll discuss why they may have been wrong in doing so. So back when the stock tanked, in March of 2019, it was because they had conducted a futility analysis and stopped the development of the drug based on that analysis. Futility analysis is a statistical analysis that determines whether a study should be stopped early because the experimental arm is unlikely to outperform the control arm. So in this case, if the drug is unlikely to be better than placebo when the study is completed, then they will stop the study for futility. So the specifics of these analyses are determined before the study starts as part of the study design, and this analysis is conducted by an independent group, which is important, as we'll discuss later. And these futility analyses, the whole point of including these is just so you can save time and cost if a study is unlikely to succeed. If you have a high degree of statistical confidence that a drug will fail, you don't want to spend tens or hundreds of millions of dollars continuing a study if you know the result is very likely to be not what you want anyway. So why do they think that decision to stop the study was incorrect? So now we're just going to go into the slides that Biogen presented in October of 2019 when they made this announcement that they're going to submit this regulatory filing. I'll have a link to these slides in the description of the video so you can take a look at it yourself. We'll just go through it here. So essentially what they said was that after the futility analysis, there were two things that happened. They obtained a larger data set from the study, and then they did a new analysis on this data set. And that the new analysis of the larger data set showed that aducanumab met the primary and secondary endpoints, apparently due to more exposure to high-dose aducanumab in the larger data set. So that's interesting, but we should automatically, be a little bit skeptical of this. We shouldn't necessarily doubt these results, but we should certainly not accept them at face value. So this is a post-hoc analysis. We'll talk about later why these are, not necessarily too reliable, and we shouldn't convince ourselves that a drug works based on a post-hoc analysis. But essentially, if you're doing a analysis that's not pre-specified, doing analysis after you already have the data for a study, you're generally going to be able to find something positive if you cut the data enough. So you have to be very careful about doing those kind of analyses. So we need to be careful when we look at their analysis and data and make sure that we understand what they're doing is rigorous, and that the conclusions they draw are supported by the data and the analysis. So this slide is just another one that Biogen presents. It says a lot of the same things as the last one, but there's important additional context that Biogen here says that emerge is positive and engage is believed to be supportive. So they plan to file this drug for approval. So what does that mean? So it's important because FDA generally requires a drug is demonstrated to be safe and effective in 2 large, well-controlled studies. And in some cases, it will allow only one large, well-controlled study and then, quote-unquote, confirmatory evidence. So that's what Biogen seems to be referencing here to me, is that emerge is this large, well-controlled study that was positive, and then engage is, quote-unquote, supportive or confirmatory evidence. So FDA provides definitions and guidance for interpreting these terms in this guidance document and others listed here. It's definitely worth skimming if you're interested in learning more about the regulatory process. But essentially, this is important because Biogen is saying that there is a regulatory pathway for approval. If they had one successful and one failed study, then they couldn't really make a case for regulatory approval, right? When you submit for approval to FDA, you have to comply with their guidelines and you have to follow certain sort of predefined regulatory pathways to get approved. But if they're able to position the ENGAGE study as confirmatory instead of as a failed study, then they can try to get approval using the one positive study plus supportive evidence criteria. That doesn't necessarily mean FDA will agree with that. FDA could just say that aducanumab needs 2 positive studies, or that ENGAGE doesn't count as supportive evidence. But Biogen is saying that at least there is a path here to get approval based on the data that they have. And ultimately, the strength of Biogen's new analysis should determine how FDA views the correct regulatory pathway. So let's look at the data here. So first, we're going to discuss the results of the studies, and then we'll look at the new analysis that apparently changed Biogen's mind. So if you recall, there were 2 studies here, emerge and engage. They both had the same design, with one small exception that we'll discuss later. But they studied the same patient populations, patients with mild cognitive impairment due to Alzheimer's disease and mild dementia. They both had 3 arms, a placebo arm, a low-dose arm, and a high-dose arm. They both measured the same primary endpoint and other secondary endpoints, and there were similar size studies. So the primary endpoint, again, was CDR-SB. This is basically a validated way to measure dementia, progression of dementia and cognitive decline in Alzheimer's disease. So just a quick reminder, the ENGAGE study did not meet its primary and secondary endpoints. The results were not statistically significant, there wasn't any dose response, and there was some nominal worsening of dementia with the high dose. And if this table doesn't make sense to you, you can take a look at the previous video where we walk through what all of this means in detail. And one last note on this slide is this is the larger data set. So I mentioned earlier that Biogen, part of what made Biogen change their mind is that they had access to a larger data set. So this is that larger data set, and it still doesn't look great, but more on that later. Okay, so on the other hand, that was the ENGAGE study. The emerge study, it did look more positive. You had a statistically significant decrease in the progression of Alzheimer's as measured by CDR-SB with the high dose. Not statistically significant with the low dose, but it was still an improvement. And then you had a dose response here. You also saw similar findings with the secondary endpoints. You got statistical significance on a lot of these with the high dose, not all of them. And then you had mostly, the positive signs in terms of slowing the progression of Alzheimer's as measured by these endpoints with even the low dose. So in addition to those clinical endpoints, Biogen also presented some biomarker endpoints. So if you recall, aducanumab is based on the amyloid hypothesis of Alzheimer's disease. The hypothesis that clearing amyloid beta plaques can slow the progression of Alzheimer's disease. So this slide presents data showing a statistically significant reduction in amyloid beta in both the high and low dose arms of the emerge and engage studies. So this drug seems to be pretty effective at reducing the level of amyloid beta plaques. You see statistical significance at all doses in both studies. You see a good dose response, and this all looks as it should based on what this drug is supposed to do. So this is important for a few reasons. So one, the drug is having the intended biological activity of clearing amyloid beta. They may not be having the intended clinical effect of slowing AD, but it is having the intended biologic effect. And then two, it's important because it potentially opens the door for them to get accelerated approval. So accelerated approval is a mechanism that enables companies to get approval based on surrogate endpoints. So for some drugs where there's an urgent unmet medical need and measuring clinical endpoints may be too complicated or take too long, FDA wants a way for these really sick patients to get drugs that might work as soon as they can. So for cancer, for example, the clinical endpoint that really matters in determining whether a drug helps patients is overall survival, but it can take many years to measure that. So oftentimes, FDA will enable companies to get accelerated approval based on a surrogate endpoint like objective response rate, which correlates with the important clinical endpoints, but is often quicker to measure. So in this case, amyloid beta could potentially be a biomarker for AD progression. It could potentially be a surrogate endpoint that predicts clinical benefit. And if it is considered that, then it could support accelerated approval, where basically Biogen get approval for this drug if they show it reduces amyloid beta plaques, and they wouldn't have to necessarily show improvement in clinical endpoints. However, it's quite controversial to say that amyloid plaque reduction or amyloid beta is a biomarker or a surrogate endpoint for Alzheimer's disease. If you recall, the big weakness in the amyloid hypothesis is that level of amyloid plaques does not correlate with disease. And if you look at the data that Biogen presented here, you see that there's not really a correlation between the clinical endpoints here and the reduction of the levels of amyloid beta plaques. So yeah, it potentially opens the door for them to get accelerated approval based on this biomarker, but you would have to convince FDA that reduction in amyloid beta plaques is a surrogate endpoint. And that's not necessarily an easy sell. So then here they just also show the effect on another biomarker, tau. So here they have a phosphorylated tau on this chart, and then this is total tau in the CSF. So it looks pretty clean for the eMERGE study. It's a little bit messier for the ENGAGE study, but this is just sort of another slide that they report, and not exactly sure what the relevant conclusion is here. I mean, obviously, when ENGAGE looks good, it looks not as good. eMERGE looks good, but they do show, and this is another biomarker that is potentially predictive of the clinical effect in Alzheimer's. So this slide is basically just summarizing what we just talked about, and then it also expands a little bit on the additional data and the analysis that changed Biogen's mind. So what did Biogen see that made them change their mind and think that they actually had a real drug here? So overall, eMERGE was a positive study, with statistically significant reduction in the primary and secondary endpoints with the high dose, and ENGAGE failed on all the primary and secondary endpoints with the low and the high dose. But Biogen said that after the futility analysis, they collected data on more patients, and these additional patients had on balance more quote-unquote exposure to the drug. So what exactly that means, more exposure, and why did some patients get more exposure? We'll dig into that later. But they also mentioned that there were protocol amendments that enabled more patients to reach a high dose and have them get a high dose for a longer duration. And that the difference in the positive eMERGE study and the negative ENGAGE study are attributable to this exposure, more exposure to the higher dose. So if this is a little bit confusing, that's okay. When I read through that point, I was confused as well. And to be fair to Biogen, this is just an intro slide, just kind of a summary that lays out their argument, and then they'll explain more and present data later. But I just wanted to pause here and just mention the importance of when you're interpreting data, you need to make sure you can follow the company's line of argument. And sometimes that just means you need to do a little bit more work and homework so you can understand what the company is saying. But sometimes if you don't understand a company's argument, it means that they're not being clear or that they're skipping steps or that they're not showing all the data, and that is cause for concern. And this is especially important with post-hoc analyses like what Biogen is doing here. So once you have the data for a study, you can keep doing post-hoc analyses. You can keep doing different cuts and analyses of the data. And if the study's big enough, you can usually do enough of these cuts to get a positive result. So this is why it's so important for all studies to pre-specify their main endpoints and also to pre-specify the statistical analysis plans. So you can't just sort of data dredge and cherry pick positive findings. So whenever a company does a post-hoc analysis, you should be cautious in drawing any conclusions from it. So post-hoc analyses can be great in terms of generating hypotheses to test more rigorously in later studies, but they really should not be used to determine whether a study succeeded or a drug works. And ideally, if companies do present post-hoc analyses, they would adjust them with statistical techniques to reduce the risk of data dredging or drawing incorrect statistical conclusions. Ideally, they would also present the results of all the post-hoc analyses they did, not just the ones that looked good. And at the very least, they should provide justification for why they chose a particular analysis and explain very clearly how they did that analysis. So if you're confused, sometimes you just have to read up a little bit, but sometimes it's on the company to be very clear about what they did. And in this case, it is very much on the company to support its claims here. So let's try to allay the confusion, break down what Biogen said here. So first, we're going to define some of the terms again. So a protocol amendment, recall that Biogen mentioned the protocol amendment is part of what drove this new analysis. So a protocol amendment is basically a change to how a study is done during a study. So as we mentioned earlier, you need to predefine the study protocol. You need to predefine how a study is done and how the data are analyzed to maintain the integrity of the study. And this protocol needs to be done before the study starts. But in some cases, it does make sense to amend the protocol when the study is ongoing if you learn something new that will affect the study. So these amendments generally need to be submitted to FDA before they're implemented. So you can't just change the protocol and then tell people that you did it afterwards. And then often these are reviewed by FDA and the IRB, the Institutional Review Board, which is basically an independent group that helps to design the study, monitors the study, and then looks out for the interests of patients. So that's what Biogen did. They did a couple of protocol amendments here. And there's one that's important here. So this protocol amendment, if we go back to this slide, we see that they did an amendment to enable a certain subset of patients to get a higher dose. So the specific subset of patients are patients who are carriers of the APOE4 mutation. So this is the strongest genetic risk factor for Alzheimer's. And in this study, these patients got a lower dose than non-carriers. And so both the high-dose group, these carriers got a lower dose than the non-carriers. Same with the low-dose group. And then the protocol amendment just enabled these APOE4 carriers to get the 10 mg per kg dose, which is the same as what the non-carriers did. So I'm not sure exactly why they did this protocol amendment. It could potentially, you'd imagine, lead to an improvement in the performance of this high dose. If the drug works and you get a higher dose of it, you'd expect the results to be better. Or if the drug doesn't work, it may do nothing. And we'll look into what exactly happened with this group later. So the next important thing to understand is the timing of this protocol amendment. So the protocol amendment applied to both the ENGAGE and EMERGE studies. So the protocol amendment alone couldn't explain why ENGAGE failed and EMERGE succeeded, except for one small detail. So that small detail is that ENGAGE started 1 month before EMERGE. So because the protocol amendment happened at the same time in March 2017, because ENGAGE started earlier, they enrolled more patients prior to the protocol amendment than the EMERGE study did. So that means that the APOE4 patients in ENGAGE received the lower 6 mg per kg dose for longer than patients in EMERGE. So just to make that a little bit more clear, here we're just kind of isolating the effect of the protocol amendment. So basically, ENGAGE, the failed study, there were more patients who got this 6 mg per kg dose. So this period here before EMERGE started, while ENGAGE was ongoing, up to the protocol amendment, these APOE4 patients got a max of 6 mg per kg. So the percentage of time that these APOE4 carriers in the ENGAGE study, the percentage of time they got this lower dose compared to the higher dose is higher. So that's what they mean by exposure. So the EMERGE study, there was more exposure on a sort of relative basis to the high dose than there was in the ENGAGE study. But the details matter here. So there's only a 1 month difference between the timing of these two studies. Is how many patients got enrolled during this time? How many patients overall got this lower exposure? And also, these patients would have had at least, or at most, 1 month, it seems like, of one extra month on the low dose. So, there's definitely a scenario where that 1 month could make a difference in the study, but it could also be too small to matter. So we really need to know more details to draw any conclusions here. So the other detail that's important, the last one, is that the enrollment stopped for the full study after the enrollment stopped for the futility analysis. So they only needed to enroll a subset of patients to do this futility analysis, and they had all of those patients enrolled in June of 2017. but they continued to enroll patients until the total study was fully enrolled in July 2018. So this is the larger data set that Biogen refers to. The patients that were enrolled in this window here are included in the larger data set, and they're not included in the futility analysis. Okay, so now that we sort of understand the terms here, we understand the protocol amendments, we understand how the protocol amendment could have changed how the dose level that patients received and the potential clinical impact. So why does this matter? So this slide describes why these changes could have made the futility analysis wrong. So basically, the futility analysis is intended to measure the probability that aducanumab would have had a statistically significant improvement on the primary endpoint greater than the placebo did. So if both doses of if both dose arms of both studies have less than 20% probability to beat placebo on the primary endpoint, then they stop the studies. And this criteria was actually met in both of the studies. However, the issue arises when they say they use pooled data from both studies to do the calculations. So this is only appropriate if the studies are identical. So you can get more statistical power if you pool studies, and you're able to pool them if they're identical studies, basically. But because the timing of the protocol amendment in relation to study start was different between emerge and engage, that means the studies are not identical. And they knew that when they implemented this protocol amendment. They knew that it could have a differential effect on the two studies, but they did not anticipate the magnitude of the effect. So that's the general rationale for why the futility analysis could be wrong. The futility analysis was based on pooled data, which assumed identical study design, but this protocol amendment made the study's design not identical. And they said that there was a larger than anticipated effect. They don't actually give us any data on that. Maybe they will later, but we'll just have to take them at their word now. So speaking of data, they haven't presented any new data here. So this is still the most important data we've seen. And again, this is from Engage. This is the larger data set, and this still does not look good. So until they present new data that makes us believe otherwise, this is really all that we have to work with. So if you think about the larger data set, if you include the larger data set and it still failed, then that 1 month difference in the timing of the protocol amendment, it has a lot of explaining to do. So there must be, if there had been a lot more patients in emerge who got the 10 mg per kg dose for longer, and those patients must have done better than the patients who received only the 6 mg per kg dose, if that actually did make a big impact on the results of the primary endpoint. Okay, so how many patients were we talking about in terms of the larger versus the smaller data set? So there were 139 patients in Engage that were in the larger data set that were not in the futility analysis. So it's hard to know whether that's enough to change the interpretation of the study. We would really need to see the details of their calculations to know that. But again, this is another slide that sort of shows that there is a difference between the futility analysis and the new data set, but we don't know the impact of that difference without seeing any data. And up to this point in the presentation, Biogen hasn't actually presented any new data to show the futility analysis was wrong or to support the claim that a subset of engaged patients actually did succeed at the primary endpoint, and that you could explain the difference between emerge and engaged based on that subset of patients. So this is the first and only slide really where Biogen discusses new data from the new analysis. So this should be like the money slide. This is the one that all of this analysis has been leading up to. So just first, we'll just walk through what we have here on this slide. So on the left, these two charts show the change in amyloid levels in emerge and engage. This is similar to what we saw earlier. And again, you see a nice decrease in amyloid levels for the drug arm versus the placebo. And then on the right, we see the change in the primary endpoint CDR-SB. So this is slightly different presentation than what we saw before. So previously we just saw this in a table with the percent decrease relative to placebo in CDR-SB. And now we see the CDR-SB change from baseline for placebo versus the drug. And what we'd want to see if the drug was working is that this slope is lower than this slope, which means the cognitive decline is lower in the drug arm than the placebo arm. So the other important difference here is that we're looking at not the low dose or high dose group, but we're looking at this specific subset of patients who received aducanumab, and that subset is defined as patients who received at least 10 uninterrupted 10 mg per kg dosing intervals at steady state. And if you look at the data here, you see that it looks like they did better than the placebo patients in eMERGE, and it looks like they did slightly better in ENGAGE as well, although the difference isn't as big as in eMERGE. And because there's no significance testing, because this is a post-hoc analysis, we don't actually know if this is a statistically significant finding. There's only 97 patients here in the subset, but this is sort of what we have to work with. This is what Biogen has shown us. So this is it. This is the data, the new data they've presented, the new analysis. I don't really know what to make of it. I don't find it too convincing personally. It's a small subset of patients. This is a post-hoc analysis. I don't know whether this would be statistically significant or not. Don't really know what to make of that. They don't really show any data on specifically how this would have invalidated the futility analysis. That doesn't mean they don't have that data. They just don't show it here. Another concern is they don't show any of the secondary endpoints. You would think that they have that data and they would have done the same analysis of this subset on the secondary endpoints. And you would assume if it was positive, they would have shown it. And generally, when companies don't show data, it's not always a good sign. So that's a little bit of an open question for me. And then they also don't really define exactly what this subset is and why they chose this particular subset. So like, why did they choose, over 10 or more uninterrupted dosing intervals? Why not 8 or why not 12? They also don't really define what steady state is exactly, although maybe that's defined elsewhere. I didn't see it though. And then, what exactly is uninterrupted? What counts as an interruption? I have a lot more questions than answers at this point, at least just for me. And the reason that I feel like that is important is the point we mentioned earlier about when companies do these post-hoc analyses, you always have to default to skepticism. We also have to default to skepticism when interpreting any data, but especially post-hoc analyses on things like this where the stakes are high. And to me, it looks like this is a potentially interesting finding, but it could just be a statistical artifact. Would love to see more data, just more data from this study without even doing a new study to better understand what to make of this. But just based on this alone, I'd probably want to see another study to see if this is actually real. But it doesn't really matter what I think, obviously. It matters what the FDA thinks, it matters what physicians think, it matters what patients think. And we also have to consider that, there was one positive study with this drug, the eMERGE study. And should we prevent patients with Alzheimer's from accessing a drug that might work? So that's something we'll talk about a little bit in the next video. We're not actually going to opine on this, but we're going to review the discussion that the FDA AdCom and other groups like ICER had around this, because it is an important issue. But before we get into that topic, which will actually be in the next video, we're going to talk about some more reasons just why we should critically evaluate Biogen's data. So we'll go back to this question of why a company would submit for approval a drug that doesn't work. One reason that we just talked about is that there is data suggesting that the drug actually does work and their initial analysis was wrong. The other reason is just they have incentives to do so, right? It's just worth it to submit it and see what happens. And in this case, the risk reward for Biogen is pretty positive. So the risk, or really just the cost of submitting this is, I don't know, maybe tens of millions of dollars to put together an FDA submission, submit the document, go back and forth with FDA. So that's the cost. And then the reward is they could be the first product in $100 billion a year market. This could be like potentially one of the biggest drugs ever. So if the cost of submitting is so low and the reward is so high, then you don't really have to have a very high probability of approval to make it worthwhile, to make it make financial sense for Biogen to submit this. So it's definitely in Biogen's interest to file this drug for approval. That doesn't mean it's in society's interest. It doesn't mean it's ethical to do so. And this drug would cost the healthcare system tens of billions of dollars for potentially, no or marginal benefit. It could potentially will potentially give patients and their families false hope, and it could actually potentially harm them, which we'll talk about in the next video. But that's why regulators like FDA are important, because the financial interests of companies do not always align with the interests of patients and the healthcare system and society. That's not to say they're never aligned with the public good, and I think in general the biopharma industry is quite well aligned with the public good in comparison to other industries. mean, the fact that they spent billions of dollars developing an Alzheimer's drug, there's no other, institution in the world that does that, and I think that's great. But we do need regulators to watch out for situations where there is not alignment, and we have to critically interpret the data because, in this sort of cynical scenario where Biogen's submitting a drug that may or may not work for approval, they're not going to just say, hey, we did a futility analysis, the drug doesn't work, but we're just going to submit it anyway. They have to make some sort of case for approval. As we discussed earlier, there has to be a regulatory pathway, and then they also have to provide some clinical justification that it worked. Even if there's a regulatory pathway, you can't just submit a drug that you openly said failed for approval. That just doesn't work. So they would have to have some sort of plausible, pseudo-plausible clinical case for why this worked. And the data we just looked at is that an actual legitimate analysis of why a futility analysis did not work. Does this show that the drug actually is potentially a beneficial product for patients with Alzheimer's, or is that just Biogen trying to, make a case for approval of a drug that, may or may not actually be effective? So we already discussed this earlier, and again, we don't really know exactly what's going on here. We have to evaluate the data from the perspective of, a sort of unbiased perspective, but also keep the incentives in mind. But again, I think that we just want to basically set the stage with this video for a discussion of the broader FDA and clinical and commercial discussions of this drug that would ensue later on. So we have to be able to understand the data, we have to understand how to critically analyze the data, and then we can critically analyze others' critical analysis of the data, other people who are smarter than us, more experienced than us. And we will do that in the next video. So next video, we're going to assess the regulatory and commercial implications of the clinical data, including this big stock jump here, and then the subsequent decline of the stock price as well. So we will talk more about the interesting stuff that happened behind the scenes here in the next video.